Chapter 4 Cause and Effect. I think the fictional detective Hercule Poirot would have admired Robert Koch. With rigorous order and method, Koch was the man who finally proved beyond a reasonable doubt the guilt of the culprits behind three crimes against humanity. Koch was the man who finally convicted the first three germs of disease. Koch, his microscope, and the splenic fever. In the late 1860s, young Robert Koch graduated from a German medical school, but repaired some soldiers during the Franco-Prussian War, married, and then settled down to a quiet life as a respectable country doctor. In 1871, on his 28th birthday, Frau Koch bought her husband a microscope to play with and inadvertently launched his career as a germ detective. While Koch played and practiced with his new microscope, the sheep of Europe were dying by the flock from a mysterious ailment that would turn their blood and their spleens black. Their spleens would swell with the blackened blood until they almost seemed to fill the torso. Occasionally, shepherds would grow feverish too and break out in black sores. Sometimes they would die. Certain fields and pastures of Europe had become deadly ground. Any flock of sheep or herd of cattle that grazed upon these cursed lands would become feverish and die with blackened blood and swollen spleens. These fields could kill even if they had been untouched for years. Was there something different in the soil of these cursed fields? Something wrong with the local water supply? The local weather? This mysterious affliction went by a variety of names: splenic fever, the black disease, or in France, la charbon, the charcoal disease. Now we call it anthrax. As he attended to his patients with one disease or another, Dr. Koch would sometimes despair over his ignorance and his inability to do anything but dispense empty words and palliative pills. Even in Koch's day and age, doctors still didn't understand what really caused all of that interference with people's breathing, or their digestion, or their blood flow. People got sick, and even doctors didn't really know why. And without understanding, there was little they could do. It was very disheartening. But in his newfound hobby as a microscopist, Koch was growing in skill and technique, and he would put whatever he could find under the lens. So when anthrax broke out in the countryside around him, it was only natural that he would turn his microscope onto the blood of anthritic sheep, and he discovered strange rod-shaped things existing there. A few other doctors with microscopes had already noticed stick-shaped corpuscles in the blood of anthrax victims. They looked like a new kind of bacilli. Often they would line up end to end in long winding chains, vaguely resembling a freight train. They might make one think of infusoria, except that these were found in mammal blood instead of in water, and they didn't wiggle or move or grow or change in any way that would show they were alive. The doctors with microscopes before Koch had even guessed that these sticks might have something to do with the disease. Maybe they were a consequence of the disease, or maybe they were even the cause. But to most educated people of the time, that was a baseless and foolish guess. The French were still coming around to Pasteur's way of thinking, and elsewhere most people still thought disease was due to internal factors. How could something so invisibly tiny cause enough harm to kill a huge cow? Those bacilli that looked like freight trains were probably just some kind of bodily residue, some debris of the disease. At worst, they were just harmless colonists seeking disease blood as their natural habitat. They surely weren't the cause. Koch saw those sticks. He suspected they might be the cause of the disease, and then he set himself to the challenge of proving it one way or the other. The germ of anthrax. If these new bacilli were the true seeds or germs of splenic fever, if their presence was the cause of the fever, blackness, and death, then these bacilli should always be present in the blood of diseased animals, and they should never be found in healthy animals. Koch began to gather his evidence by searching the blood of animals, hundreds of them, and passing sample after sample after sample under his lens. In blood samples from diseased animals, as long as the disease had progressed sufficiently to show symptoms, he could usually find the strange trains of rods in the blood. In healthy animals, he never did. So far, so good. These bacilli definitely seemed to be connected to the disease somehow. But there had been no evidence yet that these sticks were actually living things. Koch never saw them move, or grow, or change in any way. Maybe they lived a motionless plant like lifestyle. If so, that would make it more difficult to demonstrate that they were alive. But if those quiet sticks in the microscope field were really living parasitic germs, capable of growing and reproducing in their host, then Koch should at least try to show that they could multiply. Furthermore, if these bacilli were the true cause of anthrax, they should turn a healthy animal into a sick one after they entered the animal for the first time. Koch needed to show that the change from a healthy animal to a sick one happened when there was one and only one change, the entry of the bacilli into the animal's blood. But here Koch faced problems. Proving that the sticks were alive and that they caused splenic fever demanded experiments, preferably a great many of them. Putting together the rest of the puzzle and building the rest of the case against the germ was going to require lab work, but there was nobody to teach Koch how to do this. Except for Pasteur, nobody in the world knew anything about measuring or nurturing microbes, and even Pasteur had not yet tried to tackle human diseases, so Koch single-handedly developed the art of handling diseases in a laboratory. Lab work. First of all, filling a laboratory with messy, noisy, hungry, diseased sheep was out of the question. Mice would be much better. But can mice be given anthrax, and how? Maybe one could poke a little diseased sheep blood into them, but then how could you be sure you weren't contaminating the mice with any other microbes from around the laboratory? The hollow hypodermic needle had been invented a couple of decades earlier, but it is unclear if Koch knew of it or if he could obtain sufficient quantities cheaply. With practice, Koch eventually worked out a reliable procedure for sterilizing wooden splinters, dipping them in arthritic blood, and poking them into mice. The mice fell ill and died. Many mice. Consistently. But had the mice actually died from anthrax or for something else? Simply being poked with a wooden splinter probably wasn't good for their health. Koch became an expert at murine autopsies. Sure enough, in the diseased mice, the blood and spleen were always black. What about the bacilli? Under the microscope went samples of blood and spleen. There were the sticks in the blood. Koch invented a way to give anthrax to mice artificially in a laboratory by contaminating them with diseased blood. He had found a useful experimental animal, and he had shown that the disease can be transmitted by something living, or at least reproducing, in the blood. And he continued to find the mystery sticks in all diseased blood and never in healthy blood. So the case for the prosecution was gaining strength, but the defense still had reasonable doubt. The bacilli had been linked to the disease, but they had not been shown to have any actual activity or measurable effect, nor even to be alive. If these sticks in the blood were living parasites, then perhaps it would be possible to nurture them and grow crops of them outside a host in their own germ aquarium of some kind. Could Koch find a way to culture the bacilli? Perhaps if he could do that, he could watch them eat and grow and reproduce. He could watch them and handle them in their own test tubes or petri dishes, rather than having to work inside a living sheep or mouse. He could observe and experiment with the parasites in vitro rather than in vivo. But how? If the bacilli are alive, what do they like to eat? Do you need to keep them warm? Koch reasoned that if they thrive in mammalian bodies, they probably prefer to be kept warm, and they might like to live in mammalian fluids. But he also needed to keep them in clear fluid so he could watch them. Anyone who has ever taught or participated in a middle school science class may be familiar with cow eyeballs. They are readily available as a byproduct of beef production and are relatively easy and fun to dissect. If you've ever dissected one, you may remember the watery fluid that leaks out when you cut into the cornea. This is the aqueous humor, and Koch realized that it might be just the clear, nutritious incubation fluid he had been looking for. He jury rigged a body temperature incubator. He placed a small piece of diseased mouse spleen into cow eyeball juice, and he placed this test sample into the incubator, and then he waited. His incubator worked too well. If there were any anthrax sticks in his culture, they were completely hidden by a jungle of other microbes that had overtaken his microbe aquarium. Apparently, microbes had traveled in from harsher conditions, thrived in the luxury environment Koch had set up for them, and crowded out the anthrax sticks. Now what to do? Koch needed to find a way to block out all other microbes from the surroundings and culture only the anthrax sticks by themselves. Perhaps Koch could have used Pasteur's swan neck flasks, but those wouldn't have made it very convenient to work with the microbes. Instead, Koch invented another very simple, ingenious solution. He placed a tiny shred of mouse spleen in a droplet of aqueous humor onto a thin glass slide, and he placed the glass slide upside down over another piece of glass with a little hollow well formed into it. The droplet hung there, stuck to the ceiling in its own closed chamber between two pieces of glass, cut off from the outside world and out of reach of dust or any other microbes. This time after incubation, the jungle contained only one species, the anthrax bacilli. After only a few hours, they had grown from a few threads clinging onto the bits of spleen into massive tangles of twine surrounding them. They still didn't appear to wiggle or breathe or grow, but in those few hours they must have multiplied, because now there were many where once there were few. They had multiplied like a living thing. Koch had known they must, because a few of them in a wooden splinter could turn into a mouseful a day later. But now he had clearly demonstrated the existence of reproduction outside the mouse under controlled laboratory conditions for the first time. Would this new generation of bacilli cause anthrax? Koch extracted a small sample and placed it not into a mouse, but into another hanging drop culture well. He incubated this and repeated the process several more times, starting each new culture from a carefully chosen sample of the previous one. He wanted to be sure he had a purebred culture of anthrax bacilli, with no trace of contamination from anything else. Finally, after 8 generations, he found himself with a purified culture of anthrax bacilli that hadn't seen living animal flesh since their ancient ancestors, and with these far descendants, he inoculated a mouse. It fell ill and died. Koch confirmed by autopsy that it had black blood and a swollen spleen, and he confirmed by microscope that the blackened blood was filled with new bacilli. The case for the prosecution was now pretty convincing. These tiny bacilli that looked like freight trains existed in all mice killed by anthrax, and they were never found in healthy mice. They could reproduce and be cultured like living things, and their introduction into a healthy mouse was followed by death by anthrax. For the first time in history, Koch had made certain that one particular kind of microbe was the direct cause of one particular kind of disease. What should Koch do next? He was a country doctor and an entirely self-trained and self-made scientist working alone. He didn't announce every new experiment, and he didn't participate in the usual back and forth of other scientists. He preferred to be rigorous and systematic, and to be sure of his convictions before he entered public debate. Sometimes having to answer the objections of others can help you make your own arguments better, but sometimes they just get in the way and waste your time. If you are clear thinking and hardworking researcher or lawyer, it might be more efficient to think of objections yourself and to answer as many of them as you can. Before you open your mouth, you anticipate possible responses and try to answer as many as you can before other people can even ask them. You take your time and you play devil's advocate for the opposition. You try to defeat your own arguments. This virtue of thoroughness and thinking ahead can be found in many of the heroes of our story, including Leeuwenhoek, who was also something of a loner, and Spallanzani, but Koch exhibited this virtue in spades. So far, he had only demonstrated the relationship in his laboratory mice, but mice might be different from sheep. Now that Koch had reliable and well-practiced procedures, he could try larger animals. He methodically repeated his disease and germ tests with guinea pigs, rabbits, and eventually sheep. No matter the animal, the bacilli and the disease of anthrax always went together as cause and effect. Now, were anthrax bacilli the true cause of the anthrax disease? Almost surely. But one credible rebuttal still remained: the cursed fields. And Koch would not be content until he found the answer. The mystery of the cursed fields. We can suppose that the living germs of anthrax, like most living parasites, leave their host at some point and find a new one. Perhaps we could explain the cursed fields by supposing they are full of anthrax bacilli deposited there by their previous host and lying in wait for the next host. But Koch's germs, if they were living things, could hardly be immortal or invulnerable. Like any living thing, they must die without food or in hostile conditions. In his laboratory, Koch had observed that his cultures weakened and disappeared when neglected too long, or when kept in the wrong conditions. If the natural environment of these anthrax bacilli was the warm, rich, comfortable blood of mammals, then how could one explain the existence of germs in a mountain field which had been free of livestock for years, and had been frozen and buried in snow and subjected to droughts, and who knows what else in the meantime? What kept the tiny parasites alive in hostile fields season after season when they died on glass in 2 days? Koch found the answer and the final piece of the puzzle in one of his failed dried up cultures. In the samples of faded and apparently dead germs, he noticed that the remains of the culture had filled with little oval speckles. Was this contamination by some other interloping microbe? Closer examination revealed that the speckles were actually inside the corpses of the former germs. Was it some sort of decomposition? Was it some other kind of microbe that ate the bacilli? Koch kept that sample for a month and noticed no change. Then he decided to see what would happen if he put fresh cow eyeball juice on these speckles. Would they dissolve and go away? Would they grow and reproduce into clouds of speckles? No, they turned back into long chains of anthrax rods. It looked like the speckles were just a different form of the anthrax bacilli. Could these reborn bacilli cause anthrax like the original pre-speckle bacilli? Yes, they could. Koch performed more experiments and found that these speckles could endure and wait out cold. They could survive heat up to a point, and they could survive drying out, and they could remain infectious for months under a range of adverse conditions. The anthrax bacilli had a trick up their sleeves, and Koch's thoroughness had just found them out. Whenever times were lean or conditions were harsh, the bacilli would package themselves for storage. They could turn into these hard, durable, inactive, hibernating speckles, and the speckles could unpack themselves back into living bacilli whenever they were returned to healthy, for bacilli, conditions. It was as if each bacillus could turn itself into its own seed and then back again whenever it needed to. Koch had discovered that some bacteria form spores. So much for the mystery of the cursed fields. They were not cursed. They were full of spores of anthrax bacilli. The public indictment. The year was 1876. Koch still couldn't name the mechanism by which the tiny germ caused damage. He couldn't explain how the life habits of the bacilli made spleen swell or blood turn black. But Koch finally felt confident enough to present his case to the public. He knew with certainty that these bacilli were the cause of the disease anthrax. He packed his bags with samples and equipment, rode a train to the prestigious University of Breslau, and delivered a 3-day lecture, complete with demonstrations, illustrations, and plenty of microscopes. He presented his case thoroughly and methodically. with overwhelming evidence and unassailable logic. He won over the audience and the scientific community at large. The doctor and self made scientist had expertly proven a cause and effect relationship. For the first time in history, Koch had securely linked a specific infectious disease to a specific microbial cause, the parasitic bacterium now known as Bacillus anthracis. The germs of human diseases. Anthrax is mostly a plague upon livestock, on herbivorous mammals, and maybe rodents. Human beings can catch it from animals, but it doesn't usually spread from person to person, nor cause epidemics among populations. It is mostly a concern of individuals who work with livestock or those who travel to strange lands. If you lived in Koch State, you were probably far more worried about the chronic, spitting, bloody cough that was slowly consuming people from the inside out across much of Europe. Consumption was the leading cause of death at the time. Depending on the decade in the nation, it was responsible for 1 in every 8 deaths, 1 in 6, sometimes 1 in 3. This is the disease that killed Emily Bronte, Frederick Chopin, and Doc Holliday. The only consolation, if you could call it that, was that the damage took hold and progressed fairly slowly, allowing people to remain functional for up to 5 years or so. But what caused it? Nobody who became afflicted with the disease ever had any idea where or when or how their affliction began. We now know that symptoms can appear months or years after infection. Was consumption due to some flaw in nutrition? Was it an inherited disease? Nobody knew. Sometimes doctors would examine the lungs of people killed by consumption, and they would find small lumps or tubercles. In 1834, the disease began to be known by a new name, tuberculosis. In 1869, a French scientist caused tuberculosis to appear in rabbits by injecting them with matter taken from diseased humans or cattle, thus showing for the first time that tuberculosis is actually transmissible and contagious. It was more than a flaw in nutrition. There was an external agent. If anthrax is a contagion caused by a living parasitic microbe spreading from host to host and infecting one after another, maybe tuberculosis is too? Following his success with anthrax, Robert Koch marshaled his new invented methods, techniques, and skills, and he redirected them to this devastation upon humanity. Tuberculosis. Again we begin. If the disease was caused by a germ, then one should be able to find that germ in every diseased animal, but one should never find it in a healthy animal. Koch managed to obtain the corpse of a man recently killed by a severe attack of tuberculosis, so he could search for a culprit microbe. But this time, there was no germ to be found. The anthrax bacilli had been fairly easy to stumble across. These tuberculosis germs, if they were there at all, were well hidden. Anyone who's ever played with a hobby microscope and searched for critters in pond water knows that the world in the window is often not very colorful. Sometimes all you see is a glowing pool of light with faint shimmery edges of shadows here and there. It can be like looking at a jellyfish or a clear glass bead underwater. Watery things swimming in water are much easier to see if you can paint them somehow. I'm not sure if Koch can be said to have invented the art of microscopic staining, but he was certainly one of the pioneers. In the previous few years, he had taught himself how to stain a variety of microbes with a variety of dyes. In the search for the culprit of consumption, Koch tried every stain and dye and combination he could, and he finally found a dye that revealed a new germ. He found a strange, artificially colored trespasser in the lungs of the dead man. This time, instead of chains of rods that looked like freight trains, he found wispy threads tinted faintly blue by his dye. As before, the hunt with the microscope now had to be followed by extensive laboratory testing. Could he give guinea pigs tuberculosis? He tried injecting them with ground-up tubercles. They died. Had they died of tuberculosis, guinea pig postmortems reveal new tubercles inside the corpses. Now for the key question. Had wispy threads appeared in the new victims? The microscope revealed new generations of threads thriving in the diseased guinea pigs. Koch was nothing if not thorough. He begged diseased tissue from hospitals and morgues across Berlin, and then he injected it into hundreds more guinea pigs, as well as mice, rats, rabbits, cats, dogs, chickens, pigeons, and a pair of groundhogs. He never found the wispy threads in healthy animals, only in animals that died. Koch had his suspect. He had a solid candidate for the germ of tuberculosis. As before, now Koch needed to isolate, culture, and purify the suspect microbe, and then inoculate an animal with these purebred cultures to see if this created tuberculosis in the animal. But again, this germ proved more truculent than the anthrax bacilli. This one did not like aqueous humor, nor would it grow in any other culture media that Koch tried. Maybe these fussy eaters needed live food, or at least fresh blood? Koch figured out a way to make jelly out of sterile blood serum, and he tried growing his suspect thread germs on it. At first, it looked disappointingly like another failure, but maybe this microbe was just an unusually slow grower. After all, tuberculosis can take years to kill a person. Koch decided to wait and see. After 2 weeks, he finally saw spots. Tiny colonies of something had grown on the blood jelly. What were they? Were these spots little cities of the suspect threads? Or were they something else? The microscope showed that they contained the wispy blue tinted threads. Koch had figured out how to care for his second microbe. He could grow this one in test tubes on blood serum jelly. His microbe husbandry expertise had grown to 2 species. Having found a suitable home for them, with microbes, food and home are much the same thing, Koch set out to isolate, to purify, and to prepare pure cultures of one and only one microbe. He collected samples from dozens of his deceased animals and set about growing crop after crop, generation after generation, each time starting one culture from an extract of the previous one. After months of purification, he was ready to try the crucial test. Would an inoculation of this purebred microbe result in the appearance of tuberculosis? As before, one animal was not enough. In another attack of thoroughness, Koch injected his purified microbe threads into guinea pigs, rabbits, hens, rats, mice, and monkeys, as well as animals not known to suffer from tuberculosis naturally: tortoises, sparrows, 5 frogs, 3 eels, and a goldfish. Animals in the naturally immune group never got sick, and the wispy threads never appeared in their blood. But the guinea pigs succumbed. It's unclear what happened to the other mammals. Different animals have different levels of susceptibility to human tuberculosis, and I suspect Koch found erratic results with the others. The guinea pigs had contracted tuberculosis, and the only change that had preceded that was the entry into their bodies of one kind, and only one kind, of microbe. Koch had satisfied another of his requirements for proof. Injecting a healthy, susceptible animal with a purebred germ culture caused it to become ill, and with that germ-specific disease. The final requirement, finding a new generation of germs in the formerly healthy animal's body, quickly followed. The microscope revealed that the next generation of germs had filled the bodies of the newly diseased rodents. The disease and death of the guinea pigs had been accompanied by the growth and reproduction of the living parasitic germs in their bodies. Koch was now certain that these wispy threads were the true cause of tuberculosis, but he decided to see if he could fill in one more piece of the puzzle before presenting his final case to the jury of the world's doctors and scientists. As with anthrax, he wanted to understand the means of transmission. How do the tuberculosis parasites move from one host to another? All of his laboratory animals had been given tuberculosis by injection, but tuberculosis is primarily a disease of the lungs. People probably caught the disease normally, outside the laboratory, by inhaling it. Could Koch give his animals tuberculosis not with a needle, but through the air? How in the world can you spray deadly germs into the air safely? Koch's inventiveness again solved the problem. He built his animals an airtight kennel with a spray nozzle and then sprayed a mist of the deadly threads into the air. Without ever touching the animals with a needle and with himself safely outside the chamber, he succeeded in killing them with nothing more than an airborne mist. In 1882, Koch, the country doctor and independent scientist, traveled to Berlin and addressed a meeting of the biggest names in German science. The next day, headlines around the world trumpeted the news. The deadliest assassin of humanity had been found. Doctors flocked to Koch to learn how to culture the germ, how to make it appear under the microscope, and how to inoculate animals with it. As with anthrax, there was still no cure, but at least the dread disease could now be effectively diagnosed, and intelligent measures could be taken to avoid it and to keep it from spreading. Robert Koch had caught the culprit of consumption, the parasitic bacterium now known as Mycobacterium tuberculosis. His thorough efforts to answer all doubts and sort out all difficulties had securely proven a cause and effect relationship a second time. He had now logically, persuasively, and permanently identified the microbial cause of two major diseases. Cholera. In the 1800s, epidemic diarrhea or Asiatic cholera simmered quietly in India with occasional sorties into neighboring lands. In 1883, outbreaks erupted in Egypt and threatened Europe. Pasteur by this time had been spurred into action by Koch's discovery of the anthrax bacillus and had shifted his full-time energies into discovering and destroying the causes of disease. When cholera erupted in Alexandria, both Germany and France sent a germ hunting team to Egypt to find the cause. Pasteur sent a couple of his assistants, and Robert Koch went himself with one of his own assistants. It was an odd sort of competition inflamed by Franco-Prussian rivalry, but also strengthened, I think, by a distant respect between two giants, fighting ultimately for the same thing. When one of the French team, a youthful newcomer named Thulier, succumbed to the deadly disease he had been studying, the German team paid their respects at the funeral. Robert Koch helped carry the casket. Eventually, Koch and his dyes revealed a tiny new bacteria in the cholera patients. This one was not a stick-shaped bacillus or a Staphylococcus, but a wiggly, comma-shaped microbe, a vibrating vibrio. With the suspect identified, Koch then traveled to India, where the disease was abundant, to attempt to prove that this was truly the germ cholera. Animals do not suffer from human cholera, and if you can't work with lab animals, the only thing you can do is go somewhere where there are many sick people. By now, Koch was an old hand at being a germ detective, and the wiggly cholera vibrio offered few of the challenges of the spore-forming anthrax germ or the hard-to-find and hard-to-grow tuberculosis germ. Koch marched through his steps of proof fairly quickly this time. He searched every victim he could find, as well as hundreds of healthy people. He always found the vibrio in diseased patients and never in healthy people, nor in any of the animals he searched, from mice to elephants. The germ was easy to grow and purify on standard beef broth jelly. The only weakness in his case this time was that Koch could not demonstrate that injection of pure cultures caused the disease, because he could not deliberately inject people. Further investigations show that the cholera vibrio is easily killed by drying out. Unlike the anthrax bacillus, it does not have the ability to transform itself into temporary weatherproof spores. However, unlike many parasites, it does not necessarily require a host. It can live just fine in water outside a body, as long as it can find water that sufficiently resembles the contents of a person's intestines. Koch showed that the water tanks in many huts were harboring the germ and that it could sometimes be transferred by soiled linen. For the parasite, this ability to live in foul water meant that it didn't have to be too aggressive about finding a new host right away. But for human civilization, it meant that cholera, unlike the spore forming anthrax or the airborne tuberculosis, could easily be stopped by basic sanitation. Modern water and sewage treatment systems have virtually exterminated cholera in developed countries. For a third time, with rigor and thoroughness, with order and method, Koch had proved a cause and effect relationship, this time between the disease of cholera and the parasitic bacterium we now call Vibrio cholerae. Koch's rules of proof. Louis Pasteur had inspired, or perhaps provoked, both Koch's discoveries to return his attentions to the war against human diseases, and Pasteur would go on to become a giant in the business of combating disease. Pasteur would eventually give humanity the means to wipe away many infectious diseases, and he saved millions of lives. Koch never provided a successful cure for anything, but Koch was king when it came to rigorous proof. Koch's conviction of those first three germs of disease wiped away confusion and debate and allowed future disease fighters to concentrate their fire where it belonged, on the microscopic parasites that cause the harm. But constructing certainty from scratch is like assembling a three-dimensional puzzle. Even with centuries of observations and experimental data to work from, earning a conviction in the court of science is still a lengthy and difficult process. Consider how careful and methodical Koch had to be to prove his case. He always started by identifying a suspect germ, an unusual microbe in a diseased patient. He then showed that this suspect microbe is always present in diseased animals and never in healthy animals, and he did this in as many examples across as wide a range of conditions as possible. He then found a way to isolate and purify this microbe and to grow purebred crops of it in some kind of organic food in a test tube. This confirms that it is a living thing and enables the next step. Perhaps the most convincing step in the proof is to prepare a pure injection of that microbe, and only that particular microbe, and show that the inoculation is followed by the appearance of the disease. The effect follows the cause. If the only thing that changed was the introduction of a particular microbe, one can be pretty certain that microbe was the physical cause of the subsequent disease. Finally, Koch would re-isolate and re-identify the microbe from the blood or the tissues of the sickened animal. This showed that the microbe has reproduced and filled the body as the body became sick. Koch didn't yet know how microbe anatomy worked, or the precise way in which germs caused the damage, but at least he could show that their reproduction corresponded to the progression of the disease. Koch always followed this methodical, rigorous procedure, and he always ended with conviction. If you are searching for truth, if you want your conclusions to be sound, if you want your quest for knowledge to end in certainty, this is what you must do. Later in life, Koch settled into government advisory work and published his rules of germ hunting in a formal way in a government report. These rules of proof later became known as Koch's postulates. As Galileo may have inherited some ideas from Renaissance thinkers before him, perhaps Koch inherited some ideas from Francis Bacon or John Stuart Mill. Koch's postulates certainly bear a resemblance to Mill's methods for identifying a cause and effect relationship. But like Galileo, Koch built his own legacy. He was responsible for the triumph of the germ theory. He was the first to prove a cause and effect relationship between specific parasitic microbes and specific infectious diseases.